Yours, Dr. Shacklett.
Okay, is this still on? Yeah, it is. Okay. Yeah, well, one of the great things about going last is that everything has already been explained very clearly, so that makes my job a whole lot easier. My lab is studying mucosal immunity to HIV in a whole spectrum of folks, and we have been doing this kind of work for at least the last 6 years since I moved to UC Davis. And prior to that, I was at UCSF, and I have a lot of great collaborators here, including in this room, from UCSF. So this is a long study, and I'll show you a little bit of our what our plans are for the next few years, as well as some of our results gained over the last 5 or 6 years. So as Doug Kwan pointed out, there's been a real renewed interest in what HIV does to the gastrointestinal tract, and I think Peter actually also alluded to this too. This was an area of great interest way back in the late 80s even. There was a group in Berlin that was called the Berlin Diarrhea and Wasting Group, which studied GI symptoms in people with AIDS. And so it was, you know, as you I think all are aware, HIV. We all know that HIV impacts the gastrointestinal tract, and originally this was considered to be, you know, a wasting disease that really takes on the GI tract early on. But for some reason, I think as with the advent of antiretroviral therapy and people living longer and living more healthy lives with living with HIV for longer periods of time, this sort of aspect of HIV infection has been kind of set aside and not really focused on until very recently. And these are some papers that were published beginning in about 2006 and 2007. Sort of, again, calling the field, you know how everything goes in waves and in fashion waves, and it's the same way in science as it is in fashion or architecture or art. Everything comes and goes in waves, and so immediately there were 1 or 2 papers followed by a whole slew of papers, and then everyone in the field seemed to sort of become interested again in HIV in the gut. But we realized, I think, that we had been overlooking this very important site of HIV pathogenesis, and in fact, people who have made their careers studying mucosal immunology in the gut can tell us that the majority of the body's T cells are really housed in the lining of the GI tract. And this is something that, for me as an immunologist, actually was kind of a new concept and really struck me. And when you think about, it's not only the majority of the body's T cells, but it's the majority of the body's antigen-experienced memory CD4 positive T cells. Now, what do we all know about CD4 positive T cells that are activated? Those are the prime target cells for HIV infection. So, of course, when HIV gets into the gut, what does it do? It rapidly wipes out all of these CD4, not all, but the vast majority of these CD4 T cells that are what we call activated, and have a memory phenotype. They express typically 2 of the major co-receptors for HIV infection, CCR5 and CXCR4, so they're perfect primed targets for HIV infection, which so this begins to explain then one reason that HIV it preferentially infects the gut regardless of the route of infection, and whether you get the virus intravenously or intrarectally or intravaginally, the virus will very quickly find your gut CD4 T cells and begin to deplete them. So, so this is a whole slew of papers, and this is taken from one of them, just showing, this is actually experimental infection of rhesus macaques with simian immunodeficiency virus, and showing the time course of CD4 T cell depletion. And over on the right, that bar graph, if you look at the black bars, this is just showing you the raw numbers of CD4 T cells in the lining of the GI tract, and starting at day 3 post-inoculation, you see that there's almost 2,000 T cells per square millimeter, but then that drops rapidly down, and as you can see, by day 21, so only 3 weeks post-infection, the number of CD4 cells in the gut has just plummeted. And this is much more dramatic than what you see in peripheral blood, even though I don't have blood on this slide to show you as a comparison. And now, something else that's very interesting, so you see that that drop-off in CD4, something else that's very interesting is nothing's happening to the CD8 cells, or in fact, actually the number of CD8 cells is actually going up in the GI tract to the point, if you look at the white bars, they start off at about 1,500 and they go up to about 2,500 by day 50. And so there is an influx. These are the CD8 T cells that Steve McGillis was talking about. These are the killer T cells that are really the effector arm of the immune system that are out to eradicate virally infected cells, help us out here. And by firing off their ammunition of perforin-containing granules and granzyme-containing granules and so forth, and secreting cytokines. So these are the soldiers that are arriving on the scene, trying to mop things up, trying to help out. And so these are the cells that my lab is really focusing on, the CD8 T cells that appear in the gut just during acute infection, but they remain in the gut at very high frequencies throughout HIV infection, throughout chronic HIV infection. So again, these studies have been hard to do because, of course, it's relatively easy to just get a needle stick in your arm, give some blood. It's much more difficult to convince somebody to come in and undergo a rectal biopsy. So what are we talking about here? We're talking about the whole gut-associated lymphoid tissue, which consists anatomically of Peyer's patches, mesenteric lymph nodes. We all know about the lymph nodes that we have in our head and neck area that become swollen during acute HIV infection. We also have lymph nodes throughout the mesentery that surrounds the gastrointestinal tract. So the GI tract is just packed with lymphoid tissue. And then in addition to all of that, just within the lining of the gut itself, just within the lining of the gastrointestinal sort of tube, we have what are called intraepithelial and lamina propria lymphocytes. And the intraepithelial sites in particular are of this CD8 positive killer T cell phenotype. And their role in life is to defend the gastrointestinal tract against infections. So this is why the whole lining of the GI tract is really very rich as an immune site. Lamina propria lymphocytes normally are CD4 cells, but as we know in HIV infection, those CD4 cells get wiped out. So what you see if you do a histology stain of somebody with chronic HIV infection is that instead of CD4 cells in the lamina propria, they have primarily CD8 cells that have come in to fight infection. I didn't bring that slide, but I probably should have. This is a slide that I use in teaching, and I borrowed this from a mucosal immunologist in Japan. I was very struck by this analogy that he used at a meeting. The size of the gastrointestinal tract, if you were to slice it open and stretch it as far as it'll go, the GI tract would span 1.5 tennis courts, 400 square meters. And that's pretty impressive to me. So the question that we're asking in my lab is, how do mucosal T cells respond to HIV infection? And so again, we're specifically focusing on this area of the body where we know HIV is very important, eradicates CD4 cells early on, but again, it's extremely difficult to study. So we do functional analyses of mucosal CD8 positive, again, killer T cells, the same cells that Steve was talking about a moment ago. And we hypothesized that the quality of mucosal T cell responses might be related to clinical status. In other words, that controllers might have better T cells, more active T cells that are CD8 T cells that are better able to kill virally infected cells and respond to infection. And we are looking at many of the same parameters that Stephen just described, although we're not able to look at proliferative capacity of gut cells because they just don't last that long in culture because of their activation status. Our second hypothesis is that genetic factors may contribute to T cell quality but may not be the whole story, and you've heard this from the other speakers this morning as well. So what do we do? What kind of torture do we put people through? Well, we like to think it's not really all that bad, and in fact, I myself have volunteered for this on a couple of occasions. So I just want to say that we take very minimally invasive biopsies. This is a protocol that we worked out in collaboration with Dr. Peter Anton, who's a well-known GI doc down at UCLA. And we're now doing the same thing in collaboration with Peter and Steve Deeks and the SCOPE study at SFGH. We take about 20 to 24 biopsies per procedure, but the good news is they're extremely tiny, as you can see in the picture on the left. With respect to the size of a nickel, they're very, very tiny. We've enrolled over 100 patients so far, no serious adverse events, And the analyses that we do are based on flow cytometry. They're very quick. We do them the following day after the biopsy procedure. We look for the secretion of various cytokines and also for the release of cytolytic granules by CD8 T cells. And this is just to show you, just to make one quick point about HLA and genetics. In our San Francisco cohort, we have an overrepresentation of women and African Americans, which is interesting. When I say overrepresentation, meaning our cohort as a whole is largely male and Caucasian, but amongst our controllers, there are more females and African Americans than one would predict from the demographics of the group as a whole. So that's fairly interesting. And our HIV controllers tend to preserve CD4 cells in the gut. So this is, I need to go through this fairly fast, but controllers, elite controllers, are over on the far left, and the first column shows you peripheral blood. The second column shows you rectal mucosa, and as you can see, rectal mucosa in the elite controllers shows good preservation of CD4 cells and significantly greater as compared to the non-controllers. I don't have a pointer, but the red boxes sort of in the middle of the graph are non-controllers who have much more CD4 depletion in the gut. And then just the last point I want to make is that we found in a study that we published earlier this year that controllers have strong polyfunctional mucosal T cell responses. Now, what does this mean translated into? I don't have a lovely drawing like Stephen's to show you, but basically we think that controllers have more abundant, on the whole, okay, because there is again variation within the group. On the whole, controllers tend to have an abundance of HIV-specific killer T cells that home to their gut and probably proliferate within the gut, as Stephen was saying, although we haven't been able to prove that because we can't do quite the same assay. In addition, we know that these cells are highly, what we call polyfunctional, meaning they're able to secrete many antiviral and pro-inflammatory cytokines simultaneously and also release cytolytic granules, which translated into plain English means that they do a lot of things well. Okay, and so we think this is one of the differences, and part of this is probably genetically determined because many of the controllers that we find with these polyfunctional responses are indeed HLA-B57 positive, but not all of them. This is a comparison with a controller group. I'll just flip through these quickly because I know time is an issue right now. Patients on heart do not, on the whole, have very strong or polyfunctional CD8 T cell populations in their gut, so that's a distinct difference. One thing that we're very excited about, this is not published yet, is that we're also finding that many of our controllers have unusually strong CD4 responses in the gut. And as you can see, the one group that stands out in the box on the right, controllers with the MHC class II allele, HLA-DRB113, tend to have much stronger mucosal CD4 responses towards HIV antigens. So we're working on this in collaboration with some other folks at UCSF and hoping to publish this soon, and we think this is another genetic locus that may be important for control. So what have we learned? Many controllers have strong and polyfunctional mucosal T cell responses, and genetic associations play a role in these responses, but are still not the whole story, and there's a lot more to learn. What's next? We have 3 NIH-funded studies in our lab, and I'd encourage any of you to ask me questions during the break. I also have our study coordinator for one of these studies, Jane Pinnell, here, and she's just going to talk for a couple of minutes about the first study on the list here, HIV in the female reproductive tract, which is a 5-year study that we're just now starting with the Women's Interagency HIV study. The Y study here in San Francisco. The other 2 studies are also great. The ongoing study that we have with Peter Hunt and Steve Deeks, Mucosal T-cell immunity, is still ongoing, and then we're starting a new study of central nervous system biomarkers in HIV controllers. And the goal of that, I'm just going to skip ahead a little bit because I know I don't have any more time, is to scan to determine the extent to which controllers have any sort of neurological involvement in HIV infection. And this, I think, I'd be happy to talk to more of you about this during the break if you're interested, but we're going to be doing MRI scans as well as lumbar punctures and either or if anyone's interested in volunteering for that. This is our research lab, And these are great people working in the lab, and you are what this is all about. So